Hello students, today I will tell you about plant photoreceptors and particularly phytochrome. So plants have different photoreceptors that can detect different wavelengths of light like UVA and UVB radiation, blue light, red light and far red light. For example, UVR8 protein is a photoreceptor that can detect UVB radiation. Cryptochrome, phototropins and zyklopy, which is a F-box protein, can detect UVA and blue wavelengths of light. Phytochromes can detect red and far red light. So in this diagram you can see that UVR8 photoreceptor can detect UVB radiation and the wavelength of UVB radiation is 280 nanometer to 320 nanometer. Cryptochromes, phototropins and zyklopy are the photoreceptors which can detect UVA radiation and blue wavelength of light. UVA radiation has a wavelength of 320 nanometer to 380 nanometer and the wavelength of blue light is 380 nanometer to 500 nanometer. Phytochromes are the photoreceptors which can detect red light and far red light. Red light has a wavelength of 620 nanometer to 700 nanometer and far red light has a wavelength of 700 nanometer to 800 nanometer. Plant photoreceptors such as phytochrome, cryptochrome and phototropins consist of a protein that is bound to the light absorbing pigment and the light absorbing pigment is known as chromophore. These chromophores have a property to absorb a particular wavelength of light. So the spectral sensitivity to plant photoreceptors is because of the chromophores. They can detect a particular wavelength of light. Phytochromes and cryptochromes are the soluble receptors and they function in the nucleus to control the activity of various transcription factors. Whereas phototropins are plasma membrane associated protein kinases. Upon the absorption of light, they cause their autophosphorylation and induce the signal transduction pathways. Now we come to the first photoreceptor, that is phytochrome. Phytochrome was discovered as a photoreversible pigment present in plants by Hendricks and Bothwick. For the first time, phytochrome pigment was identified using spectrophotometer by Butler and Seigelman. The name phytochrome was also given by Butler. Peter Quayle and Clark Lagarius chemically purified the intact phytochrome photoreceptor molecule. First phytochrome gene sequence was published by Howard Hershey and Peter Quayle in 1985. Phytochromes are mainly found in plants where they affect various physiological processes such as seed germination, seedling development, shade avoidance response in plants, flowering and circadian rhythm regulation. Circadian rhythm in plants refer to biological cycle that lasts around 24 hours governing various physiological and metabolic processes. These include activities like leaf movements, stomatal opening and closing, opening of the flower petals and closing of the flower petals, photosynthesis, hormone production, etc. Phytochromes absorb red and far red light of the visible spectrum and phytochrome-like proteins have also been detected in fungi, algae and bacteria. Photoreversible response of phytochrome was observed during lettuce seed germination. It was seen that when soaked lettuce seeds were grown in dark, they were unable to germinate. But when red light was given, the seeds germinated. If the red light was followed by far red light, there was inhibition in seed germination. So it was observed that seed germination depended on the last treatment given to the seeds. If the last treatment of light was red, the seeds germinated, and if the last treatment of light that was given was far red, it showed inhibition of seed germination. So this experiment showed that red light promoted seed germination in lettuce, whereas far red light inhibited seed germination. Phytochrome exists in two interconvertible forms. The red light absorbing form of phytochrome is known as phytochrome red, and the far red light absorbing form of phytochrome is known as phytochrome far red. Phytochrome far red is the active form of phytochrome. PFR is again converted to PR after absorbing far red light, or under darkness PFR form converts into PR form. Since the red light and far red light spectrum overlaps, so phytochrome pool is never fully converted into PFR or PR form. When broad spectrum far red light is given, PFR is converted into PR. But at photostationary state, only 98% PR is present and 2% PFR is present. Now we come to the molecular structure of phytochrome. Phytochrome is a soluble protein photoreceptor. It is a homodimer. It means it is made up of two identical subunits. The molecular mass of phytochrome is 250 kilodaltons. The protein part of phytochrome is known as apoprotein, and the light absorbing chromophore is known as phytochromobilin. When phytochromobilin attaches to the apoprotein, the holoprotein or complete phytochrome is formed. Phytochromobilin is the light absorbing chromophore. It absorbs red light or far red light. Phytochromobilin is a linear tetrapyrrole. It is synthesized inside the plastids from heme. Phytochromobilin attaches to the phytochrome apoprotein in the cytosol through a thioether linkage to a cysteine residue, cysteine amino acid. Phytochrome apoprotein has multiple domains. For example, we have nose, ears, eyes, mouth for different functions. Similarly, phytochrome apoprotein has different domains which have particular functions, including chromophore binding, dimerization, or signal transduction. When PR is converted into PFR, it causes conformational change and unfolding of N-terminal from C-terminal. In this diagram, you can see that phytochromobilin is synthesized inside the plastid from heme. It comes out in the cytosol where it attaches to the apoprotein part of the phytochrome. The phytochrome has NH2 terminal or N terminal, and on the other side you can see COOH written, that is the C terminal. The GAF domain of phytochrome apoprotein is the region where phytochromobilin attaches with the thioether linkage. The PRD domain of the phytochrome apoprotein has the nuclear localization sequence, and the histidine kinase like related domain is found in bacterial phytochromes where it has a bacterial two-component sensor regulator system. When PR form absorbs red light, the D ring of the phytochromobilin undergoes change from cis to trans form, and when this D ring rotates, it causes the unfolding of N terminal from C terminal. Thereby, the nuclear localization sequence in the PRD domain are exposed, and this active form of PFR goes into the nucleus where it activates the transcription factor, thereby causing gene expression and expressing the morphological response. Phytochrome also has different forms like phytochrome A, B, C, D and E. The phytochrome A form requires the protein FHY, which provides the nuclear localization sequences, and then it carries the phytochrome A into the nucleus. Phytochrome controls various photomorphogenic responses such as phytochrome regulates seed germination, red light promotes seed germination whereas far red light inhibits seed germination. Phytochromes also control seedling growth, leaf expansion, stem elongation, phototropism, that is movement of plant part towards light, and de-etiolation means when the dark grown seedling is transferred to light, then chlorophyll starts synthesizing, it turns green. Phytochrome also influence the timing of flowering in plants. For example, in photoperiodism, I have told you that long day plants flower in long day conditions. During long days, PR form is converted into PFR, and PFR is the physiologically active form which promotes flowering in long day plants. Remember that PFR promotes flowering in long day plants whereas PFR form inhibits flowering in short day plants. Phytochromes also influence stomatal opening and closure according to light intensity and quality. Phytochromes mediate shade avoidance response. Through phytochromes, plants detect shade and the plant parts compete for the light under shaded condition. Phytochromes also regulate circadian rhythm and influence the timing of various physiological processes such as stomatal opening or closing, photosynthesis and gene expression. Now some important questions about photoreceptors and phytochrome. Name the different photoreceptors found in plants. What do you understand by photoreversibility of phytochromes? Describe the molecular structure of phytochrome. Describe the role of phytochrome in plants. And describe the molecular mechanism of phytochrome controlled responses in plants. Please like, share, comment and subscribe. Thank you so much.